Okay, so you can see this is the active site. So this darker green area is the rest of the lysozyme protein. So you can see glutamic acid and aspartic acid here. These are able to interact with the substrate. This is one sugar making up the polysaccharide and a second sugar in the chain of polysaccharides. And it attacks, so the oxygen, the charged oxygen of the amino acid, this is part of the lysozyme now, attacks the C1 carbon in the sugar, creating a temporary covalent bond. And then water comes in to finish the hydrolysis so that the product of the two smaller sugars can be released. Okay, so water is donating hydrogen back to glutamic acid, and then the hydroxyl group is being added to the sugar. Okay, so these three major steps are what's happening in the active site. You got your enzyme substrate binding, the activated intermediate, and then your product. Okay, and then the enzyme, once the product is released, the enzyme can just go back, and if there's more substrate present, go back and catalyze the same reaction again. So remember that it's the key interactions, both in this case covalent interaction and hydrolysis reaction, that are completing that chemical reaction. Okay, and this wouldn't happen at a very, very slow rate if the carbohydrate was just sitting in the cytosol, where there's plenty of water available, but it wouldn't be in that proximity to other amino acids, such that the initial step of breaking, creating a new covalent bond between the C1 carbon and the oxygen of the next sugar would be would occur. So the enzyme is basically speeding up that reaction by holding the carbohydrate in close proximity to those key side chains. This part of chapter 4, yeah, you should know the lysozyme, the function of lysozyme for our next test. Yes. So I won't ask you the detail, to remember the detail of which carbon is being attacked, but I will expect you to be able to explain these three key steps. Okay? So chapter 7 is talking about gene expression. We've skipped chapters 5 and 6, which spend a lot of time on DNA replication and mutations and repair, the fidelity of DNA. So I'm going to quickly go over a little bit of background information that you'll find in chapter 5 and that hopefully you've already learned, so this should sound familiar from general biology. So there are a few key aspects of DNA that you need to understand before we can talk about gene expression. All right, so I want you to think back. Why is DNA important? Where is it found in the cell? Kind of these basic traits that are common to all eukaryotic cells. We're going to focus on eukaryotic cells. I'll mention a couple of differences between prokaryotes and eukaryotes, but basically genes have the same, or DNA has the same function, whether it's a prokaryotic cell or a eukaryotic cell, right? So what's the purpose of the DNA molecule? Yep. Okay, so it can code for RNA or proteins. Yeah. So the best definition is that it holds and carries all of the genetic information that is necessary for the life of that cell. And as multicellular organisms, all of our cells hold the genetic information for sustaining our life as a multicellular organism. Okay, so it's a storage form. It's not really useful in that storage form. It's useful when it's converting that information into a useful form, either RNA or more commonly proteins, right? So the proteins are what do the work in the cell. So gene expression is this process of converting the information in DNA to protein. Right, so that's a subsequent step after the polypeptide. So it's storing the information that you need to make the polypeptide, which will then fold into the protein. But the end product, the useful part, is the properly folded protein. So I just want to go over some definitions that you should be familiar with. And I won't get through all of this today, but we'll go over DNA and chromosomes, and we'll see how far we get from there. So in the big picture of things, DNA is storing information to get proteins in the cell. And we're going to talk about this process of transcription and translation that are the two steps that are necessary for that conversion. But first, let's just talk about DNA. So what is the difference between a gene and a genome? Right? All of the collective genes collected together that are part of a single organism. Okay? So the human genome, which has now been sequenced and studied, it's all of that information in all of our chromosomes, which number how many for humans? 46 chromosomes. DNA also has other regions besides the genes. In fact, only about 2% of the human genome is actually been identified as genes. There's a lot of stuff in between these genes that are encoding for the protein in our genome. And right now we're calling it junk, but really I think in your lifetime we'll find out that it's not really junk at all. But we just haven't figured out what it's doing yet, or all of what it's doing yet. So we have about 20,000 or just over 20,000 genes that are encoded for in our genome. And the genome is the DNA molecule. So let's go over the structure. Let's see if I have a slide for this. Okay, so what are the building blocks of DNA? Nucleotides, right. So there's a base, nucleotide base, and a sugar molecule, deoxyribose, that come together to form a nucleotide, the basic unit of nucleic acids, right? So this is the monomer that come together to form a polymer that we know of as nucleic acid, DNA, or in the case of ribonucleic acids, RNA. So what about these bases? Here they're shown 4 of them. Are there more than 4 bases in DNA? Nope, just 4. And the way that we read the strands is from the 5' end to the 3' end. That's the convention. And what do we know about a mature DNA molecule? Is this mature molecule? Okay, here's a mature molecule. What's the difference? Okay, there's an alpha helix, but before that, what do you notice? There are two strands. Okay, there's always two strands in DNA. It's a double helix, and they run anti-parallel. And so what holds those two strands together? Hydrogen bonding. So non-covalent bonding, but it's really tightly holding them together because of what we call base pairing. So what are the bases that pair together? Purines and pyrimidines. So adenine, thymine, guanine, cytosine. Okay, those are the ones that pair together. And part of the fidelity of DNA is maintained by having this duplication of the sequence of these bases. So our cells, and it goes into more detail in chapters 5 about that fidelity, how the heredity is maintained by fixing any errors in the replication of DNA. But basically, part of this base pairing is really important for keeping that information correct. So, because we know what happens when there's a mistake in the gene, what does it affect? It affects the protein. It can be a silent mutation, but more likely it's going to affect the downstream product, the protein. And then you know if there's a change in the protein, it's going to change the structure. It's going to maybe alter the function or just make it non-functional, right? Yes, that's true. Can be a silent mutation. So usually we find DNA in the cell in a condensed form because it's actually quite a long molecule. Oh, one other thing I wanted to point out about the hydrogen bonding. What do you notice? It's drawn in pink in these cartoons of the structure. So what do you notice about the hydrogen bonding? Okay. So that when we look in a moment at the structure of these nucleotide bases, you'll see that there are 3 opportunities for hydrogen bonding between G and C and 2 between A and T. So the DNA structure is a phosphate at one end, the base at another end. The way it's oriented in DNA is you have a phosphate backbone. Okay, so covalent bonds forming between one base and the next base. The phosphates on the outside of the DNA molecules, the bases are oriented on the inside of the double helix so that they can be in close proximity to facilitate that. So within a single strand, you're getting covalent links, right? So the polymer of each strand is covalently linked. It's the double helix, the 2 antiparallel strands that are not covalently linked, but just non-covalently by these hydrogen bonds. Why is that important? Oh, we'll get to that in a minute because you don't see the other strand here. But it would be between this base and the base of the antiparallel strand of DNA because it's a double helix, 2 strands of DNA. But why would it be important for these 2 antiparallel strands to be able to break apart? Right, so replication of the DNA, transcription of the DNA to the message so that we can make protein from it, can only depend on the coding strand, only one of these two strands. So you don't want it to be covalently bonded because it would take too much energy to break it apart in order to carry out these essential functions for life. All life reproduces, right? Okay, so here are the nitrogenous bases, the purines and the pyrimidines. Remember that guanine cytosine are able to hydrogen bond in 3 places, and thymine and adenine in just 2, which I'll show you in a moment. So it's between the nitrogen group and the hydrogen groups on the adenine and the oxygen and the hydrogen on the thymine. You get those 2 covalent bonds because of the way that they're oriented. So this is where you'd find the phosphate bonds that link each of these bases. And the way that they're oriented towards the center of the double helix brings these atoms in close enough proximity so that they can have hydrogen bonding occur. Whereas in the cytosine and guanine, you get 3 opportunities to make those hydrogen bonds, either through an oxygen and a hydrogen atom or a nitrogen and a hydrogen atom. So remember that they're anti-parallel. Only one of these strands is the coding strand when we're talking about the central dogma converting this information in these genes into the form of proteins. The 3' end can be distinguished from the 5' end because there's a free phosphate group on the 5' end. So it's chemically distinct. And the bonds that we call these between the sugars are the phosphodiester bonds. So the double helix is a form that you find DNA in inside the cells, but there's even more information that we understand about the genome. So each cell has the entire collection of genes. And what form do we find that DNA in the cells? We have chromosomes. When do you really see the chromosomes? Okay, normally you don't see them, but when do you see the chromosomes? Right. Mm-hmm. Mitosis and meiosis, when they start to separate and have to be ordered and organized before so that each daughter cell gets a complete set of chromosomes. But usually in the nucleus, if you just look under the microscope, like we did for some of the microscopy work we did in lab, you can just see a dark spot inside the cell. That is less organized than when you have the process of cell division. And it's really still condensed because those molecules of DNA would be way too long to fit in a cell if they were really all stretched out end by end. It's really the chromatin that is the form that you see in the nucleus. Okay, so this condensed form that we call chromatin, it's actually not just DNA. It's DNA wound around histones, the nucleosomes. The histones that make up the nucleosomes help it to keep organized and tightly condensed, except for when some kind of gene expression needs to occur. So, each cell type has the complete genome. So, what's the difference between a liver cell and a skin cell? Yep. Genes that are expressed, exactly. Anything more? Okay. Yep, so only, at any one time, only a subset of all the genes in the genome are actually being expressed. It takes a lot of energy to go through this process of converting the information in a gene to a protein. And so different genes are turned on in different cells. And we'll talk about that later when we get to the control of gene expression in Chapter 8. Okay, so here are a couple images to go with what we just talked about. It's condensed into chromosomes. Within the chromosome, you can actually, when you stain the chromosomes, you can see a banding pattern that accounts for different genes on the different alleles. Being that we are sexual reproducing organisms, we actually have a complete set of genes from our mother and our father, so we have 23 pairs, chromosome pairs. So let's take a closer look at what genes are and what we understand about some of this intervening sequence of DNA that's important for genes and gene expression. So here in this figure you can see this is a bacterial part of a bacterial chromosome. You can see that the genes are organized on each strand in an anti-parallel manner. Often in prokaryotes, such as E. coli, the genes are clustered together, genes with common function, so that you would get an RNA transcript for 2 or more genes at one time. But in the case of eukaryotic organisms, you really only see one gene being transcribed at a time. And that first step of forming proteins is transcription, where the DNA, the information in one, the coding strand of DNA, is converted to RNA in the form of, so what are the 3 types of RNA? Transfer, ribosomal, and messenger RNA. Okay? I think I have those on a slide here. Yep, I'll just write it. So the 3 types of RNA are tRNA, transfer RNA, important, we'll talk about later when we talk about the function of the ribosome, ribosomal RNA, which is actually part of the structure of the ribosome organelle, and the messenger RNA. Okay, most of the genes in the cell encode for messenger RNA. So one thing to note about this transition of information from the DNA to the RNA is now you still have a nucleic acid molecule that the information is stored in, right? But what's the difference between the DNA and the RNA? Now you can notice right off the bat in this. Right. So RNA is single-stranded, and that's important for the next step of the central dogma of converting that messenger RNA, at least, into protein, into a sequence of amino acids. Right, so you can have hydrogen bonding within an RNA molecule, and that's what gives the T shape of a tRNA. But for the most part, it's not going to have hydrogen bonding. Right. And RNA being single-stranded is actually much less stable in the cell. It's rapidly degraded. DNA, not so easily degraded. We have RNases on our fingers that, any time that we're working in the lab with RNA, it's just much more challenging to handle RNA than it would be for DNA. So the RNA is this intermediate between the information in the gene and the protein, and the process of converting that information to a useful form is gene expression. In the case, as I mentioned, in prokaryotes, you can also often have many genes simultaneously transcribed into a single mRNA and then into their respective proteins. And then for the eukaryotes, that's actually just usually one gene per mRNA. There's a fourth kind of RNA that I'm not going to spend a lot of time on called small nuclear RNA, the snRNA. So I really want you to focus on the players that are involved in gene expression, the central dogma of gene expression, and those are the mRNA, tRNA, and rRNA. So where's the DNA found in eukaryotic cells again? In the nucleus. What about prokaryotic cells? Simpler. Not the nucleolus, that's actually a region within the nucleus. Nucleoid, which is it a membrane-bound organelle? No, it's not. It's simply a region where the bacteria store, or the prokaryote stores its DNA still in a condensed form, like we find it in the nucleus. So I'm going to focus on the more complex process of gene expression in eukaryotes, because you have to think not only about the process of converting DNA to a protein, but where things are happening in the cell. So proteins are made on ribosomes. Where do we find ribosomes in the cell? In the cytosol. Okay, so you got to have an extra piece of information in that transcript to get that RNA out of the nucleus into the cytosol, where then it can be made into a protein. Okay, so transcription, let's just focus on that first. Some of the differences between the DNA, that's the gene, and the RNA are the sugars that form the backbone of this molecule. So we have DNA deoxynucleic acid, RNA ribonucleic acid, and one key difference in nucleotide base. Okay, there's no thymine in RNA, no uracil in DNA. You can see really there's only the loss of the methyl group here. They're very similar in their structure, and their uracil is still able to form how many hydrogen bonds? And with what other base would it be bonding? Adenine, right. 2. Right, so it's still 2, like thymine and adenine, but it's uracil that's interacting with. But it's not in the RNA. The methyl group is gone, and so you don't need that hydrogen bonding, because the RNA is just a single-stranded molecule. Yeah, the only difference in the structure between these sugars is that thymine has a methyl group here at, I'm not sure what number carbon that is. Okay, so RNA also has important parts of it that are not coding regions, are not the information that's going to make up the sequence of amino acids. Similarly, DNA has these extra regions that are, in some cases, just upstream, so just prior to the gene, are important for initiating this process of transcription. And downstream, the last codon, or the last information at the end of a gene, has to tell the enzyme to stop, where to stop, right? So the terminator. So we call these two regions that are common to all genes the promoter and the terminator, and then the gene is the intervening sequence of nucleotides. So how does RNA synthesis begin? In order to make messenger RNA, you need an enzyme, a rather large enzyme called RNA polymerase. That RNA polymerase is able to break apart that hydrogen bonding between the two antiparallel strands of the double helix of DNA with the help of a factor called sigma factor, which attaches to the a region within the promoter just before the start site of the gene. Then one of the strands is used as a coding strand to make a single strand of RNA, here depicted as a blue line. And then this enzyme will fall off of the DNA double helix at the terminator. Okay. So here in this, so the template strand is always the 3' to 5'. So RNA polymerase always reads DNA in the 3' to 5' direction, making a strand that's actually what? Identical or complementary to that template strand? Complementary. So this blue strand of RNA is now matching the coding strand of the gene, not the template strand. Okay, so it has, except for the uracil replacing the thymine. So once you have the completed RNA chain, in bacteria, the ribosomes can immediately attach because this is happening in the cytosol, but in the case of the eukaryotic cells, it needs to be then moved from the nucleus to the cytosol, where it can find the ribosome to continue the process.